Hello, everybody. My name is Magdalena Gajdošová. I am from Charles University in Prague, and the ongoing project I will tell you about now is rather evolutionary oriented. There is evidence that some freshwater taxa, such as gammarids, are molecularly very diverse in the Western Carpathians, while they remain quite uniform when you cross the boundary of the adjacent Bohemian Massif. Hence, it has been suggested that the Western Carpathians served as glacial refugium for freshwater animals, which resulted in outstanding lineage diversity in this area. The main question we are focusing on is, if we analyze the molecular diversity of comparable communities in both areas using metabarcoding, will we see the same pattern, that is, much larger, perhaps cryptic diversity in the Carpathians? The community we chose as a model for Illumina sequencing for cytochrome oxidase is macrozoobenthos of spring fens. This is very convenient for several reasons. There are many rather immobile and specialist taxa, which could even enforce the diversification in the past. Also, spring fens are found in both areas we study, and the very same spring fens, and by that I mean the same sites, not only the kind of habitat, has been studied in detail for many years by our colleagues from Brno. Therefore, we know a lot about what has been morphologically determined in these spring fens, and we can compare the so far taxonomic knowledge with what we find with CO1 metabarcoding. Now, we do not have the data from Bohemian Massif yet, so we cannot answer the question I mentioned. However, we have already analyzed the data from the Carpathians, and they seem to be rich and interesting just as they are. We analyzed 21 spring fens and obtained over 8 million reads, which we filtered, analyzed, and compared to reference databases. And here is what could be interesting to some of you. When using the 97% identity threshold, we found almost 800 OTUs, out of which 57% could not be assigned to any reference barcode using bold or blast. There are two possible explanations for this. A, we encountered substantial cryptic diversity, which is in agreement with our hypothesis. Or B, there is a big gap concerning Carpathian aquatic invertebrates in the barcoding reference databases, which is yet to be covered. Of course, it is probably a combination of both, and one of the questions for the future is which of these two reasons is prevailing. Either way, this is a big piece of unknown, which is waiting to be analyzed further. We compared the OTU numbers to checklists of species known from the localities, and it is clear that the high unknown diversity is typical for several taxa, especially dipterans like tabanids and ceratopogonids, but we also found some interesting patterns in annelids like Eisenella tetraedra, which seems to comprise of 12 OTUs, or the leech Helopdella stagnalis, of which we found 5 OTUs, all of them at one locality. As you can see here, we are collecting samples of these species from more localities, so we can study them further. So all of this is just a hint that there is beautiful diversity waiting to be studied in Carpathian benthos. And if you have suggestions, comments, or questions about it, I will be happy to talk to you later. Thank you.